Hello everyone, and welcome to today's DFHCC Connect Science seminar. I'm delighted to introduce our speaker, Dr. Peter Campbell. Peter is Head of Cancer Genetics and Genomics at the Wellcome Trust Sanger Institute in Hinxton, Cambridge, UK, having joined the Cancer Genome Project there in 2007. Before that, he completed training in hematology in New Zealand and Australia in 2002. And in 2006, he was awarded a PhD by the University of Cambridge for his research in the molecular pathogenesis of myeloproliferative disorders. He was made a Wellcome Trust Senior Clinical Fellow in 2010. Peter has pioneered efforts to decode the patterns of mutation and structural variation in the cancer genome. His research has had a major impact on our understanding of the mechanisms of cancer evolution. In 2011, for example, he discovered the phenomenon of chromothripsis, a clustered rearrangement resulting from a one-off cellular crisis, providing evidence that cancer evolution can be punctuated by sudden catastrophic rearrangements. In recent years, he's led the analysis of somatic structural variation in human cancer, orchestrated by the Pan-Cancer Analysis of Whole Genomes International Consortium, which published its findings in a series of highly influential papers last year. I'd like to remind you to consult the Connect Science website, both to see what's coming up in future presentations and also to watch previous presentations which are available there. For today's seminar, please write your questions in the Q&A box, not in the chat box. So we're going to be consulting the Q&A box for questions. Make sure you record your questions there. So without any further ado, I'd like to welcome Peter, whose talk today is entitled Somatic Mutations in Normal Tissues.
Thanks very much, Ralph. And it's a pleasure to be here. I'm just sorry that it has to be virtual and not in person. But hopefully, as the vaccine rolls out in our respective countries, we can begin to dream of meeting up again. So what I want to do today is just cover a few things around somatic mutations in normal tissues, and in particular, the way that we can use them to as naturally occurring clonal marks that effectively define clones within human tissues, and then what that then tells us about clonal dynamics, and in particular stem cell dynamics. And to do that, I'm just going to give you a bit of background about how we can use these marks to make inferences about clonal structure, and some basic data around the accumulation of somatic mutations across one's lifespan. And then I'm going to spend most of the talk looking at some data where we've studied stem cell dynamics in prostate. This is unpublished data. And then a quick gallop across some of the other organ systems that we've applied these sorts of approaches to give you a sense of the sort of collective of the findings that we're making. So, I mean, I really don't need for this audience to dwell on this particular slide. It's you'll see this in any basic textbook of cancer evolution. But just to say that when we sequence a cancer genome at the time of diagnosis here, what we find is that the tumor is made up of a series of clones, and those clones can all trace their ancestry back through a series of cell divisions to a cell, the most recent common ancestor, that's the kind of the cell that seeds all of the current extant cancer cells within that tumor. And then from then on back, we're really pretty limited in what we can say about the evolution of the cancer before the emergence of this most recent common ancestor, because all of the cells share all of the mutations accumulated during that time. And so we have really very little insight into that very early cancer evolution process. So one of the things that we've been increasingly interested in recent years is looking at normal tissues and looking at what's happening in terms of mutation accumulation in normal tissues and in terms of positive selection and clonal evolution within normal tissues. To do that, there are a number of technical challenges that we need to overcome. So here is what 1 square centimeter of aged sun-exposed skin looks like, and each of the circles here represents a separate independent clone. And you can see that in that 1 square centimeter of clone, there are many hundreds, potentially thousands of different clones. And so you can imagine that if you mushed up that square centimeter of skin and sequenced it, what you would be doing is sequencing molecules of DNA that come from all of these different independent clones. And so if you want to find the somatic mutations in any of those clones, you're really only going to be ever sequencing 1 or 2 molecules from that clone, and that's going to make it quite challenging to identify the somatic mutations. So we've taken a couple of approaches to overcome this technical barrier. One is to use laser capture microdissection and essentially shrink down the size of the tissue that we sequence. And we can now routinely generate sequencing libraries from as few as 100 to 200 somatic cells. And that then becomes around about the size of one of these clones. So if we microdissect a couple of 100 cells and sequence it, then we are seeing all of the molecules of DNA essentially can come from 1 or 2 clones, and it makes it much more straightforward to find the somatic mutations. The other approach that we've taken is to take single cell suspensions of the tissue and then isolate them as single cells and expand them in vitro into organoids or colonies that we then sequence. And essentially the progeny of that colony all come from one cell, so they all share the mutations that original cell had. And that too has given us very robust catalogues of somatic mutations from normal tissues. We've not spent so much time doing direct single cell sequencing. In our hands, the whole genome amplification that's required introduces too many errors for identifying somatic mutations in normal tissues. I know that a number of groups, including at Dana Farber, have been more successful than us in doing that, and that approach is certainly valid if you do get good whole genome amplification without the introduction of too many errors. And there's some really exciting developments that I think will continue to advance that particular approach as well. But for the purposes of today, we've not done any direct single cell sequencing. It's either single cell derived colonies or this laser capture microdissection approach. In many ways, once one has the catalog of mutations, we're really interested in kind of using that to construct the kind of history of that clone and that tissue that we're looking at. And the way that we do that is essentially by looking at the patterns of sharing of mutations across cells. So here's a toy lineage tree shown here where the cells have undergone a couple of divisions each, and during those cell divisions, each of those cell divisions, mutations have been accumulated. And because the genome is a big place and the mutation rate isn't that high, those mutations tend to land in unique places and therefore mark that lineage forevermore with that particular mutation. And so we can take the pattern of mutations that we see in these cells and then from that reconstruct essentially a phylogenetic tree, which is much like any family tree that you see. It basically traces the relationships amongst different somatic lineages. So with that background, I'm going to kind of show some data. So the first thing to say is that somatic mutations occur in pretty much all of the tissues that we've looked at a constant rate with age. So, and what that means is that the mutation burden linearly increases with age. Now, what we find is that the rate of that mutation accumulation differs from organ to organ. So the lowest rates we found are in testes, where it's only a handful of mutations every decade. And the highest rates we've seen are in colon, where we're seeing something like 50+ mutations in every colon cell every year. But most tissues are in this kind of range of somewhere between 15 and 30 or 40 mutations per cell per year. And it is very clock-like through childhood, adulthood, and old age. We don't really see any change in the mutation rate for a given tissue across this. What's really interesting is that now we have some new methodologies that allow us to look at post-mitotic tissues and the mutation burden in post-mitotic tissues. And we find that even in post-mitotic tissues, actually the mutation rate continues at a constant linear rate throughout life, even after those cells have stopped dividing. The other thing to note is that the somatic mutations start right from conception. So here's some data where we've taken 150 blood stem cells from a 59-year-old man, and we've isolated those cells as single cells, and then expanded them into colonies and the phylogenetic tree in the usual way. And what we find is that the 150 cells essentially can be partitioned into two groups of cells, and by two mutations. Essentially, there's one mutation here and one mutation here, and every blood cell has one or other of these mutations. No blood cell has both mutations, and no blood cell has neither of them. So essentially, every blood cell has one or other of those mutations. And what we find, what that means is that essentially there was a cell here that divided to give two daughter cells, and one daughter cell got the mutation here on the right, and the other daughter cell got the mutation here on the left. And essentially that cell that divided must have been the most recent common ancestor of all blood cells in this man. Now, it turns out that when we look at those two mutations in buccal epithelial cells, we see exactly the same two mutations present in buccal epithelial cells, and at exactly the same proportions that we see them in the blood cells. So 1/3 of all of the buccal epithelial cells carry this mutation from the left, and 2/3 of the buccal cells carry this mutation from the right. So what that means is that not only is the most recent common ancestor, is this cell that divided the most recent common ancestor of all blood cells, but it's also the most recent common ancestor of all buccal epithelial cells. And it turns out that buccal epithelium is derived from ectoderm, and blood is derived from mesoderm. So this most recent common ancestor must have predated gastrulation. So we're looking right back into very early embryogenesis. And in fact, we believe that this cell, we've got more recent data where we've looked at more at this, using this approach across fetal tissues. There's a paper on bioRxiv where we've shown this data, where we've looked at multiple tissues, including placenta. And we can see the same pattern there, where we see this very early kind of bifurcation in the phylogenetic tree, with this kind of split of 1/3, 2/3. And we believe, therefore, that this, even in the placenta, and therefore that this is going as far back as the first cell division of the zygote. In other words, where even from the first cell division of the zygote, mutations are being laid down in the genome that we can use to do lineage tracing. So, and as you imagine, as you go further down the phylogenetic tree, essentially you're looking at kind of more deeper and deeper into those kind of first few cell divisions of the embryo. And essentially you're kind of, how far into that you can go basically is entirely dictated by how many cells you're willing to spend doing genome sequencing on. So with that, what's kind of useful about that in terms for reconstructing clonal structure within somatic tissues is that the mutation rate is sufficiently frequent in tissues, starts very early in life, and is a kind of constant clock-like rate, which means that we can make pretty robust inferences about the kind of patterns of clonal dynamics from the shapes and structures of phylogenetic trees. So to illustrate that in quite some detail, I'm going to show you some data from human prostate, where basically what we did was we took a single prostate, an entire prostate from a 59-year-old man, and we basically, so a walnut, a prostate's about the size of a walnut, and we essentially sliced it all the way through from the top to the bottom. And put each of those slices on a slide, and so that's about 1,200 slides, and we then imaged them and traced individual ductal units. So the prostate is made up of about 24 to 30 separate ducts that all kind of start at the urethra and then kind of invade into the sort of prostate tissue and kind of have this, each has a sort of tree-like structure, it kind of branches and branches and branches until you get to the terminal acini. And so what we did was we took a couple of ducts or ductal units from out of the 24 to 30, and we essentially tracked all of the branches of those ducts from the urethra all the way into the peripheral prostate, and then essentially did our laser capture microdissections. You can see here. So here are some of the acinal and ductal structures that we see, and then that's what it looks like after we've cut out the bit. And then these all get sent off for whole genome sequencing, and we build the phylogenetic tree. As for the other organs, we find that mutations accumulate linearly with age in prostate at around about, I think, 15 or so mutations per year on average. So that again means we can use the mutation rate as a molecular clock. So I won't bore you with how we reconstruct the phylogenetic tree. We use methods that many people have developed over the years for looking at whole cancer genomes, for reconstructing phylogenetic trees from whole cancer genomes. We essentially apply those here to rebuild the phylogenetic tree. I guess that the main point is that we find clusters of mutations. So each of the microdissections that we've made here is a column, and each of the rows is a cluster of mutations, and the brightness represents how high that particular microdissection has, how much of that cluster is represented in each individual microdissection. And you can see the sort of nested structure where there are clusters with sets of clusters of mutations which are shared across many microdissections, and then nested within that are other clusters which are seen in ever fewer numbers of microdissections, and that means that we can pretty robustly build a phylogenetic tree. This nesting is essentially means that you can construct a perfect phylogenetic tree. And so these are the two ductal units that we reconstruct from this 59-year-old man's prostate, and this is the sort of phylogenetic tree that we get back from around about, I think, about 400 or 500 whole genome microdissections. And what you can see here is that is the sort of branching structure that you would expect in a phylogenetic tree. Unlike a cancer, what you would typically see in a cancer is a single trunk with late branching, and that's because all of the cancer cells derive from this one ancestral cell, the most recent common ancestor of the cancer, and all of the mutations present in that ancestral cell represent the sort of mutations displayed on the trunk of the phylogenetic tree. When we look at normal tissue, because normal tissue is polyclonal, we don't see that same single trunk. What we see are lots of trunks, if you like, representing completely independent clones of cells. So in some ways, this kind of looks like a comb more than a tree. But nonetheless, there is still branching structure within this. So there are still kind of microdissections that are clonally related to one another. And when we look at the positions of these branch points, we see that they kind of fall into two clusters, perhaps most easily seen here on the right. We can see, and essentially everywhere that there's a solid circle represents a branch point in the phylogenetic tree. And you can see that the branch points occur at kind of cluster in two regions. One very high up, or with very few mutations accumulated right at the kind of base of the phylogenetic tree, and then a second group of branch points in the phylogenetic tree occurring at around about 300 to 400 mutations of molecular time. So because we know that mutations accumulate at a constant rate throughout life, and we know what that rate is, we can essentially use the number of mutations present at the timings of these branch points to work out how old, when during this person's life those branch points occurred. And you can see that these occur when there's hardly any mutations. And in fact, we believe that there's sort of no more than 5 to 10 mutations when these first branch points are occurring, and that very squarely dates to fetal development. And then the second group of branch points in the phylogenetic tree is occurring at around about 300 to 400 mutations of molecular time, and that's works out to be around about when this man would have been 13 to 15, 16 years old, so right during adolescence. So perhaps, not surprisingly, much of the branching in this phylogenetic tree is kind of restricted to these two time points during fetal development and then during pubertal maturation of the prostate. Now, it's worth noting that a branch point in a phylogenetic tree here essentially is a record of a historic stem cell division. So essentially a cell divided, and we are now 10, 15, 40 years later sequencing to both progeny, eventual descendants of both of those original daughter cells from that cell division. So that's what a branch point in a somatic lineage like this really implies. It's a sort of record of a historic cell division. So what this cluster of branch points tells us at these two different time points is that there would have been an increased rate of cell division during fetal development and again during adolescence. And we know from the anatomical studies of the prostate that what happens in fetal development is that there's a lot of active cell division as the kind of rudimentary ducts are laid down in the kind of developing prostate. And so presumably these cell divisions here represent the kind of laying down of the early rudimentary prostatic ducts. And then during adolescence, with the burst of androgens that occurs during adolescence, what happens is that there's further generation of further complexity within the prostate. And the formation of new ductal side branches, new terminal acini, increased branching complexity essentially. And so again, that is presumably associated with increased rates of cell division that we're now seeing in the phylogenetic tree. But we can go a little bit further than just throwing up the phylogenetic tree and talking about what this means, because we actually know where each of these microdissections sits in the prostate. And so we can now take those prostate the microdissections that we've made, and we know where they were in the prostate, and we know how they're connected to one another, how the ducts essentially are spreading and expanding through the prostate and branching and eventually finishing in terminal acini. And so we can essentially, for each microdissection, we know its physical location and the connections, what the ductal connections are. And this is essentially a two-dimensional projection of that three-dimensional branching structure. So this is one of the ductal units that we've tracked. And what you can see here, so here where the asterisk is where that duct reaches the urethra, and then you can see the branches that represent the branches of the duct as it goes out into the peripheral prostate gland, and each of the circles here represents one of the microdissections that we've made. And we've colored the microdissections for each one of the major branches of the phylogenetic tree that relates to this duct. So here's one of those major branches that's seeded by a single embryonic cell. Here's the second one in pink. That comes from a single embryonic cell, etc. And what you can see is that for any one of these embryonic cells, that there are a series of these microdissections that essentially derive from that, that have contribution of descendants of that original embryonic cell. So, and the sort of intensity of the color from white through to dark brown in the top left one here, is essentially the fraction of cells within that microdissection that trace their lineage back to that embryonic stem cell. And so what you can see is that when we look at the kind of pattern across the whole of this, this duct, is that the whole duct is essentially derives from probably a handful of original embryonic cells, no more than 5 or 10 or so. And the scope of each of these embryonic cells is largely mutually exclusive. In other words, regions where you see one embryonic cell, you tend not to see the other embryonic cells so much, although there are occasional regions, some exemplified with the arrows here, where we see a microdissection that's contributed to by more than one embryonic cell. But largely their territories, the scope of these embryonic clones is mutually exclusive. And what's kind of interesting is that there are discontinuities in the contribution of these embryonic clones. So you'll see kind of regions like this one here in the pink at the top left that are really quite discontinuous from other regions that are also contributed to by this embryonic cell. So it's a sort of complex, mixed pattern of contribution from the embryonic cells. When we look at the pubertal clones, we see a kind of similar pattern, although it's not the contribution of any given pubertal cell is not as widespread as we see with the embryonic clones. So here, for example, is a couple of branch points within the phylogenetic tree that we time to puberty. And when we look, what we see is that there's a kind of series of progressive branch points here. And when we look at the contribution, so the embryonic cell here is shown on the top right, that's cluster 61. And you can see it's had the same spread, there's kind of wide embryonic spread typical of an embryonic contribution. And then when we drill down the progressive branches that are occurring during puberty, what we see is that there are kind of individual side branches and terminal branches that are contributed to by individual cells that existed during puberty. And in fact, entire side branches like this one here for cluster 73 and this one here for cluster 103, that's an entire side branch that's seeded by a single cell from puberty. So we believe, and that cell basically makes no contribution to any other bit of this man's prostate epithelium. So it's like this one cell at puberty has generated an entire side duct, but not contributed anywhere else in that person's prostate. So it seems like puberty is driving formation of new side and terminal branches by local and very rapid expansion of epithelial progenitors that are already scattered throughout that rudimentary ductal tree. Then when we look at adult clones, we find that the adult clones are really very limited in their geographic scope. So here, for example, is a series of embryonic clones that derive from one embryonic clone. Cluster 76 is the embryonic clone here with its wide spread throughout the ductal unit as shown previously. But when we take the individual adult clones here, we see that they're spread over 1 or 2 or maybe 3 at most microdissections. And when they're in more than one microdissections, they tend to be physically very close together. And so what that suggests is that the territory of adult stem cells within the prostate is really very constricted. And probably, although we don't know this for sure, probably cell turnover is relatively quiescent and infrequent, although we're really more confident about saying that the geographical scope does not expand that much. So quite a different picture in kind of normal adult tissue maintenance than we see during development and during puberty. The one exception to that was a clone here that we saw where the branch points were definitely dated to adulthood, and the clone had spread over quite a large number of microdissections in quite a large physical distance. And what was interesting about this clone was it was the only one in the entire data set where we could find a confident driver mutation. And this is a canonical mutation seen in prostate cancer in a well-known prostate cancer gene, FOXA1. And FOXA1 is known to have hotspot missense mutations, and indeed this is one of the hotspot mutations. So this is a bona fide driver mutation that we're seeing in normal prostate epithelium, and it has driven some sort of clonal expansion within the prostate that's distinctly unusual compared to all of the other clones within this man's prostate epithelium. So I think that's really where I want to finish the prostate stuff, just to say that each of the 24 to 30 ductal units in a prostate is laid down by a handful of embryonic cells, probably 5 to 10, and that those 5 to 10 embryonic cells lay down the rudimentary ductal structure during fetal development. And then it sits in sort of limbo until puberty, and then the burst of androgens that all adolescent boys have drives the formation of new side and terminal branches alongside all of the behavioral problems, drives formation of new side and terminal branches within the prostate gland. And those side and terminal branches are seeded fairly monoclonally by local stem cells that are scattered throughout the ductal tree, acting on those local stem cells, essentially instructing them to make new branches and increase the complexity of the ductal tree. Then it all kind of goes back to quietness. Tissue maintenance in adulthood seems to be undertaken by local progenitor cells. The geographic scope of those clones is really very limited. And driver mutations are rare, but when they do occur, seem to be able to trigger more extensive clonal expansion that kind of follows the ductal tree. The clones are expanding along the ductal tree and respecting that tissue architecture. So in the kind of last 10-15 minutes, I just want to quickly go through some of the insights that we can get when we look at other organ systems that kind of follow the sort of pattern of inferences that we can make in the prostate. When we look at blood, I've shown you the top of this phylogenetic tree before, but when we look at blood, we see this kind of hugely polyclonal pattern, and we can use the kind of timings of these coalescences to infer historic population size. And the reason we're able to do this in blood is because blood is relatively well mixed, and so these represent a random sample, if you like, of cell divisions within the hematopoietic stem cell system, and that means we can use population genetics methods to infer population size. And what you see in blood, and we've repeated this now in a number of individuals, is this kind of increased clustering of branch points very early in the phylogenetic tree, which represents the expanding population size during fetal development and childhood growth. But then by the time of kind of late childhood, early adolescence, the population size hits a plateau, and these kind of branch points in the phylogenetic tree that represent historic self-renewing stem cell divisions are really there to maintain a fairly constant population size throughout life. What's interesting is that as we look at this in older individuals, we're beginning to see that that clones are beginning to expand as they acquire driver mutations earlier in life. In colon, what we see, so when we look at normal colon, we see what we can do is dissect individual colon crypts. So here's a colon crypt that's been micro-dissected and sequenced. And what we see in healthy colon is that the phylogenetic tree, like we see in blood and like I showed in prostate, is really much like a comb, that the individual crypts are basically unrelated to one another. Occasionally, we'll see a branch point in the phylogenetic tree, and which would imply that two crypts derive from the same common ancestor, and so therefore that crypt has undergone a fission event, so one crypt becoming two. But that's pretty uncommon in normal healthy colon. It's mostly those crypts are essentially evolving independently of one another and not sharing any genetic information. In contrast, when we look at colon that has been affected by inflammatory bowel disease, where in this case ulcerative colitis, which is kind of essentially strips off the lining, the epithelium of the colon, denudes it, and then eventually recovers either with treatment or through spontaneous remission. What we find is a very different pattern to the phylogenetic tree. So here are a couple of phylogenetic trees. Here's a region of bowel that we know was affected in the past by inflammatory bowel disease. And we've made a number of micro dissections along quite a long stretch of bowel here. And what we find is that almost all of these crypts actually derive from a common ancestor here that had about a couple of thousand mutations. So each of these crypts essentially it derives from the same original cell, quite a different pattern to what we see in healthy colon, and suggests that in recovering from an inflammatory insult in the form of IBD, that actually there's quite a huge latent stem cell potential within the colon that means that that entire kind of centimeter long stretches of colon can all be regenerated from a single ancestral stem cell. It's a similar pattern in liver, actually. So when we look at normal liver, we find that even very closely related, even closely physically close regions of the liver basically share very little in the way of clonal structure. You can see that this phylogenetic tree here on the left really does look like a comb. There's almost no sharing of mutations. Each of these clones is completely independent and unrelated to all of the other ones. But when we look at liver cirrhosis, and here's an example with liver cirrhosis, where we see the bands of fibrosis surrounding regenerative hepatic nodules, we see a lot more complexity in the phylogenetic tree with a lot more branching. And indeed, entire cirrhotic nodules can derive, that can be millimeters in size, can derive from a single ancestral cell. So it's much the same sort of pattern that we see in colon, that in healthy liver, there's not much, seemingly not much cell division going on. The clones are relatively small in size and lots of very highly polyclonal with no sharing of mutations. But in the presence of disease with kind of ongoing damage to the liver and then progressive damage and regeneration, that over time you get this expansion of clones that will contain many mutations, and those clones can grow to be quite large in size. And what's interesting is that each of these cirrhotic nodules, we don't see sharing of mutations across the cirrhotic nodules. So the fibrosis is essentially walling off those clones into isolated pockets, and they're evolving independently in each of those cirrhotic nodules. Quite a dynamic process. In esophagus, and indeed in many squamous tissues, one of the patterns that we're seeing is that we see this increasing rate of driver mutations with age. And so this is a sort of kind of depiction of what esophagus looks like as we age. And as I said before, the circles here represent individual clones within the tissue. And the size of the circle represents the size of the clone. And if it has a color in it, those clones carry a driver mutation, and the types of driver mutations are shown here by what color they are. And so what you can see is that there's two things going on as we get older. So in young individuals, there are certainly a few driver mutations. So this is 1 square centimeter of esophagus. Each of these squares represents a square centimeter of esophagus. And there are already, in people in their 20s and 30s, there are driver mutations present. But the number of those driver mutations spectacularly increases over time. And we believe that in most tissues that we've looked at, that the rate of driver mutations is again linear over time. So the occurrence of driver mutations is occurring pretty steadily throughout life. And so the number of driver mutations is therefore much higher in an elderly person than in a younger person. But there's a second thing going on, which is that not only are the number of driver mutations increasing linearly with time, but actually the clones themselves are expanding over time, and particularly in the presence of driver mutations. So whereas you're mostly dominated by relatively small clones in people in their 20s to 30s, and therefore the tissue's really quite polyclonal, by the time you get to people in their 50s to 70s, some of those clones have really grown to be quite large, and it's a much more, I mean, it's still highly polyclonal, but it's less polyclonal than before. And it's the clones with driver mutations that, because they've got a driver mutation, they have a selective advantage, and so they're expanding more quickly than the normal clones. And so you have this double whammy, if you like, of not only are the number of driver mutations increasing with age, but also the size of those clones is increasing with age. So that when you look at the fraction of cells within the esophagus that have a driver mutation, that is increasing exponentially with age, at least supralinearly with age. And so whereas only a small fraction of cells have a driver mutation as in when they're 20s to 30s, by the time you're in your 50s, 60s, and 70s, you could well be in a position where you've got 75 to 90% of your esophageal epithelial cells having at least one driver mutation. And indeed, in some of these clones, you can see that there are 2 or 3 driver mutations within that clone. So already these cells have taken quite a large leap on the path towards getting the required complement of driver mutations to become transformed. Finally, what we see in lung is really quite a fascinating pattern. So as you might imagine, the kind of pattern of mutations that we see in lung is dominated by whether someone has smoked or not. And in people who've smoked, we see that the mutation burden here, the black line represents the kind of maximum number of mutations that we would expect in a lung cell from a never smoker. And when we look at the mutation burden in current smokers here in the purple colors, pretty much all of the cells that we look at, all of the clones that we look at in a current smoker's lung have an increased mutation burden, often 3 or 4 times more mutations than we would expect for a never smoker. And those mutations have the sort of mutational pattern that you would expect for the mutagens in cigarette smoke. In contrast, what's really interesting is that when we look at lung from ex-smokers, we see this kind of bimodal distribution of mutation burden. And there is a population in many of these ex-smokers, there's a population of cells that essentially has the same elevated mutation burden that we see in the current smokers, but a second population of cells that essentially has a normal mutation burden, exactly the same mutation burden that you would see in a non-smoker. And what's really interesting is that population of cells is many times more frequent. We estimate about 4 times more, that population size is 4 times as large in ex-smokers than in current smokers. You can see there are occasional cells in the current smokers with a normal mutation burden, but the population is much larger in ex-smokers. And so somehow these cells have managed to evade all those decades of cigarette smoking and all of the carcinogens in those cigarette smoking. And it seems that when people stop smoking, that those cells somehow expand and eventually kind of slowly replace the cells that have been damaged by the cigarette smoke. And so this kind of, there seems to be some we believe, sort of niche that harbors cells and protects them from exposure to cigarette smoke, and from there the bronchial epithelium can kind of partially regenerate with a much essentially a normal mutation burden. And what we see when we look at the phylogenetic trees in these individuals, so when we look at a smoker, this would be a relatively typical pattern where pretty much all of the cells have this massively increased mutation burden. We see lots and lots of driver mutations throughout the phylogenetic tree, as you would expect. and you see kind of quite large clonal expansions. You can see this entire clonal expansion here driven by this p53 mutation, a second p53 mutation here also causing a clonal expansion. That's the sort of typical pattern that we would see in a current smoker. What you see in the ex-smokers is here's the sort of quite obvious population, the two populations of cells here with elevated mutation burden, 4,000 to 6,000 mutations, and here's this population of cells with a near normal mutation burden, more like 1,500 to 2,000 mutations. And this expansion of cells with a normal mutation burden is polyclonal. So it's a polyclonal stem cell expansion that's driving this population of cells with near normal mutation burden. The driver mutation rate is much lower, much less frequent in this population of cells with normal mutation burden. And it is tempting to believe that this population of cells with a near normal mutation burden is at least part of the reason why when you stop smoking, your rate of lung cancer drops away. Because we know that when you sequence lung cancers in patients who are ex-smokers. So if you sequence lung cancers from ex-smokers, and Matthew Marsden and others have done a lot of this, what you see is that almost all of those lung cancers that emerge in non-smokers do have an elevated mutation burden. So those lung cancers must be coming from this population of cells that has been damaged by the cigarette smoke, not from this population of cells that's got a low mutation burden. And so one is tempted to believe that this population of cells is in some way cancer, it has have a lower rate of cancer transformation than this population of cells with elevated mutation burden. So that's really where I want to finish today. The sort of key findings, I guess, to take away are that spontaneous somatic mutations do enable us to do lineage tracing, and we can really get quite rich insights into sort of the dynamics of stem cells and the numbers of stem cells across tissues. And we're seeing differences in the determined by, for example, physical structure. So you can sort of see the difference in squamous epithelium like the esophagus, where you've got lots and lots of quite small clones, versus a glandular tissue like the prostate that I showed you, versus the sort of typical pattern that we see in the colon crypts that I showed you. And these dynamics are really, I mean, these stem cell kind of populations are really very dynamic, and what we see is that disease, toxicity, and driver mutations, all of those things can unmask this very large regenerative potential in many tissues. So that the colon and the liver that I showed, where we see these kind of massive clones, really quite large clones, expanding in response to a disease or toxic process. Similarly in the lung, as they regenerate after when someone stops smoking. But what we don't really know is what fraction of cells and solid tissues carry this regenerative potential. Is this a large population of cells, or is this a relatively small number? And I think that remains an unanswered question, and is presumably quite important for all sorts of things around the cancer stem cell field, but also around regenerative medicine that would be quite interesting to explore in greater detail as we go forward. So just to sort of summarize the things that I've talked about over the talk, I won't dwell on that in any great detail, just to sign off by thanking the people who did all the work in this, particularly Sebastian Grossman and Yvette Hooks, and my collaborator from Newcastle, Rakesh here, who drove the prostate work. Kenichi Yoshida drove the lung work. Henry Lee Six drove the blood and the colon work. Simon, the liver work. Kate, the lung work with Kenichi, and Sigagir, the inflammatory bowel disease work, and my long-term colleagues at the same institute, Inigo, Phil, and Mike. Thanks very much.
Thanks, Peter. That was a wonderful presentation and very thought-provoking. We've got quite a few questions, so I will go straight into them if you're ready for that.
Yes, of course.
So two very closely related questions, one from Andre Nussenzweig, one from Mina Kussi, and it relates to the comment you made, I think, early on about the mutation rates in post-mitotic cells appearing to be similar to those of cycling cells. And the question is, are there any clues as to whether the mechanism is similar, or are there distinct features of the dividing versus the non-dividing cells?
So our best way of trying to infer what the mechanism of mutations is has been to look at mutational signatures. I didn't particularly cover that in any of these talks, but it seems actually that when we look at the kind of pattern of mutations, the signatures of mutations, actually they're quite similar in post-mitotic tissues to dividing tissues. There are differences. There are differences in the rates, and there are kind of differences in which mutational signatures are more active versus less active. But the signatures are present. So as far as we can tell, it seems to be the same sorts of mutational processes active in post-mitotic tissues as we see in dividing tissues.
Okay, thanks. A question from Jim Haber. Are the mutations you're tracing principally base pair substitutions or plus or minus indels? Do you detect gross chromosomal rearrangements, and how does that frequency compare to those of the simplest base pair changes?
Yes, so we can. I mean, with whole genome sequencing, we can trace all types of somatic mutation. In terms of lineage tracing, it's certainly easiest to work from single base substitution. So much of the data that I showed today was based on single base substitutions. What we find is that indels occur at about an eighth to a tenth of the rate of base substitutions, but they also occur at a kind of linear, constant rate throughout life. So indels accumulate at around about one tenth of the rate of substitution. So maybe 1 or 2 indels per cell per year throughout life for most organ systems, a bit higher in colon. Structural variation it can take a number of forms, and we do see it's much less frequent in normal tissues. But we do see occasional clones with structural variation, and we have seen, those patterns of structural variation include whole chromosome gains and losses from time to time, but also deletions, translocations occasionally, tangentifications, and even particularly in liver and in diseased liver, we're seeing occasional instances of chromothripsis and other really quite complex structural abnormalities. So structural variation does occur in normal tissues, but it's at a considerably lower rate than we see, for example, in cancers. It's probably, if one had to kind of put one's finger on the major difference between cancer and normal tissues, it's the kind of burden of structural variation for most solid tumors.
Okay. Question from Steve Elledge about ulcerative colitis and the changes in the colonic epithelium that you see there. With the expansions, he's asking, are there also more mutations in the crypts that you see expanded? He's interested particularly in the role of inflammation and the possibility that there may be Applebeck-driven patterns of mutation. So the question again is, do you see a shift in the mutation signatures in the ulcerative colitis and inflammatory bowel disease?
Yes,
crypts?
It's a good question, and so there's sort of 2 bits to this, which I think both are quite interesting. So we do see an increased mutation burden in inflammatory bowel disease for an age, compared to what we would expect for an age-matched normal tissue, and that increase in mutation burden is actually not particularly driven by Applebeck, but seems to be a kind of just an acceleration of the mutational processes that are present in normal colon, maybe because of the increased cell division rate or other kind of impacts on DNA repair. Occasionally we see the signatures of treatment as well increasing the mutation burden. So occasionally there seem like there are some mutational processes that can be attributed to particular treatments that the person's had for inflammatory disease. But most of the increase in mutation burden seems to be driven by the disease process. And we see a similar sort of thing in liver, actually, that in the presence of liver disease, the mutation rate goes up. for either as a result of the disease process or the kind of regenerative response to that disease process. Now, the APOBEC question is really interesting, because what we see when we look across all of the normal tissues at the kind of patterns of APOBEC mutagenesis is that some tissues don't have any APOBEC mutagenesis at all, and other tissues have quite a lot of it. So salivary gland and lung have a reasonable amount of APOBEC mutagenesis. But it seems to be very episodic and periodic. Bladder also has this kind of pattern. In fact, bladder is really interesting, because what you find in bladder is that you can sequence kind of regions of the bladder, and you'll have completely independent clones, and all of them will have, you know, an extra 1,000 or 2 mutations from APOBEC, and they'll all be quite physically clustered. And then, you know, go a centimeter further around the bladder and sequence a whole bunch of clones, and there'll be 0 APOBEC in those. So it seems like there's in bladder that there's this kind of regional activity of APOBEC that is not clone specific. It seems to be clone independent, and the tempting thing to believe is that there might be kind of viruses or other local insults that are triggering those independent clones all to switch on APOBEC mutagenesis, but in this quite episodic way, such that, you know, a neighboring clone a little bit further away just has 0 activity.
Okay, thanks. Question from Raymond Monat. Whether there is any data on how germline DNA repair deficiencies influence tissue-level somatic mutation frequencies or tissue clonal architecture?
Yeah, we've actually just started looking at this in a collaboration with Uri Tabori from Toronto, who's collected a tissue bank from individuals born with biallelic mismatch repair deficiency. So these are, you know, Lynch syndrome is where you inherit a single allele loss of a mismatch repair gene, and then the tumor that emerges has somatically lost the other allele. Well, because Lynch syndrome is common enough in the background population, occasionally both parents will have Lynch syndrome, and then their child has a risk of inheriting both faulty alleles, and therefore they're essentially conceived without any mismatch repair capacity. And we've now looked at normal tissues in a handful of these individuals. Unfortunately, they're not so common, but we've looked in about 8 individuals with different biallelic mismatch repair deficiencies. And what we find is that the mutation, it's really interesting, because we can look at normal tissues and we can look at transformed tissues from the same individual. And what we find is that in the normal tissues, in the absence of mismatch repair, the mutation rate is something like 8 to 10 mutations per cell per day higher than for normal for people with intact mismatch repair, suggesting that the mismatch repair pathway is repairing about 8 to 10 lesions per cell in every cell per day. And that, therefore, you know, you can imagine if you play that out over 20, 30 years, that leads to these really spectacular mutation burdens. And what's interesting beyond that is that then when you look at transformed clones from these same individuals, you see that there's an even further acceleration in the mutation burden. So mismatch repair is repairing 8 to 10 lesions a day in every normal cell. But when a cell gets the sort of mitotic pressure as it begins to transform, accumulates those driver mutations, presumably the accelerated division rate further increases the mutation rate, and then mismatch repair would normally be repairing many, many more lesions per day in those transformed cells. And so, you know, we're seeing massive mutation burdens in these tumors once the cell begins to transform. So yes, it's really quite interesting, and it seems to be we see differences. We see differences across. We see differences in the patterns of mutation between different different mismatch repair genes, and we see some kind of components of the mismatch repair pathway seem to preferentially repair particular DNA lesions. Also, those signatures change between normal and transformed cells. So transformation changes the types of lesions that are causing mutations in, presumably the transformation is introducing new lesions that otherwise wouldn't be seen in the normal cells. And we're seeing differences between tissues in the patterns of mutations. So some tissues seem to have kind of particular mutational signatures in the absence of mismatch repair, whereas other tissues will have a different complement of signatures. So really quite a complex picture whereby the tissue type matters, the genotype matters, and whether the cell is normal or transformed matters for both the rates of mutations and the signatures that emerge.
Okay, thanks. Question from Lee Ellis. If it's the cell that retains stem cell traits that is transformed in the prostate, is that primary prostate cancer most likely to undergo metastasis and be lethal to the patient? I don't know if you have any data.
Yeah, I think that's really interesting. You know, we've not one of the sort of deficiencies. One of the things that I'd like to do in the next generation of these studies is to kind of link back from and come back to patients with cancer and look at the normal tissues and the cancer in the same individual and try and understand, try and use kind of, and I think that spatial methods might be really important here. There are ways that one can in a spatial way kind of identify which cells in a tissue section have a given mutation. Admittedly, it has to be transcribed, obviously, because we have to do this with RNA-based methods. But one of the things I'd love to be able to kind of do is to kind of take a cancer from someone, and indeed a metastatic cancer, identify the mutations, and then kind of look at where those mutations are in relation to the normal tissue, and kind of see whether there are whether you see. You can imagine that in the prostate, with that one clone that had the drive mutation that had expanded over quite a large chunk of the duct, what you can imagine is that when if that had eventually transformed into a cancer, that you might be able to see the kind of stages of progression as that clone went from normal cell through clone with a driver mutation expanding throughout the ductal tree, through to beginning to generate pockets of microinvasion, through to the kind of final fully invasive tumor that then metastasizes. And I'd love to work out ways that we can kind of begin to do that. And we're sort of fiddling around with these methods, making slow progress, but they're still quite a bit temperamental for us. But I think it's a really fascinating possibility to explore.
So we're getting towards the end of time. We're going to keep going through the questions. I just want to apologize to people who've asked questions in the Q&A box if we don't get to reach your question in time. Your question will be forwarded to Peter afterwards so that he can reply. So apologies that we may well not be able to reach the end of our list. Next question is from Francisco Real. Have you been able to compare mutation occurrence in tissues from offspring of smokers versus non-smokers for women who were smoking or not at the time of pregnancy?
That's a good question. We have not done that. So there has been work done on, particularly on trio sequencing, where people have sequenced the mother, the father, and the child, the offspring, and what you see in those studies is that and this is not work that I've been involved in, but a number of groups have done this, is that there's a kind of paternal age effect of mutation accumulation. I don't think there's much evidence for an effect of maternal smoking. Most of the kind of de novo mutations in the offspring come from the father. But actually, Francesco's question is a bit more subtle than that, because he's asking actually if the in utero environment of cigarette smoking is introducing mutations in the embryo. That's a really interesting question, actually. We should do that study. That would be fun. That would be quite interesting, because smoking definitely, those carcinogens could cross the placenta, I believe. Certainly there are effects on the vasculature in the placenta in smokers. That's why there's a higher rate of intrauterine growth retardation in mothers who smoke. And that could cause problems both in the placenta in terms of the mutational pattern, but also in the fetus. That would be an interesting study, actually. And we could do that from cord blood. We can successfully sequence cord blood derived colonies and get very good mutation catalogs. So yeah, that would be an interesting study.
Great. So we have reached, I think, the point where we need to close. And I do apologize again we have, I think, 4 questions that we haven't been able to get to. So I'd just like to take this opportunity to thank you, Peter, for a wonderful, stimulating talk and for your fantastic work on revealing the origins of human cancer. So see you again soon, we hope.
Okay, see you. Thank you.
Bye.